Okay, with that, sit back and relax, buckle your seatbelts, and we're going to run through the pathophysiology, some of the new drugs, and how are we going to position all these alternative agents in our clinical practice. So as we move towards the next generation of therapeutics, and this slide is representative of dermatology, rheumatology, and GI, we need to consider the genetic factors, what's going on in the environment, and we're very interested, although very neonatal, in our examination of the microbiome in IBD, and of course how the individual's immune response, which is going now to be related to both genetics and what's going on in the environment. So first off, what's going on in IBD in the world? Well, these are relatively new diseases. Crohn's disease was not described until the 1940s. There's been history of colitis and ulcerative colitis going back many decades. But as you watch the epidemiology, and I would bet dollars to donuts, it's the same for RA or for psoriasis. When we look at the worldwide prevalence, and this is before World War II, subsequently up to 1970, we see expansion primarily in first world countries. And then as we get into the 1990s and beyond, we're actually seeing it evolve in Asia and in countries where we didn't see this before. Now, this is too fast for the genetics to change. And I will tell you that the gene mutations that have been recognized in the Caucasian population of North America and Western Europe, particularly the NOD2 or CARD15 gene, are not the same as we see in Asia or in Africa. Yet, the phenotypes of the diseases are the same whether or not it was from a Caucasian population or an early or newly evolving, for instance, Asian population. There may be subtle differences. There's some suggestion that African Americans may have more complicated perianal disease, but none of this has really been borne out across the world. So it's clearly more than genetics that are changing the epidemiology of these diseases. And again, along with RA and with psoriasis, what we see is an increasing incidence as we see decreasing risks of other infectious complications. The best protective way of preventing IBD is being born and raised in the tropics, getting infectious diarrhea when you're young, and those seem to be protective of developing these diseases. An interesting experiment done actually at UCSF, looking at exposure to animals. We know that firstborn are more likely to develop asthma than subsequent kids. What they did was they looked at dander exposure in an animal model of asthma. And so they took the dander, the dust, out of homes with furry pets and fed it to mice. And actually that protected against asthma compared to dust from homes without furry pets. And you could actually isolate microbial products that are going to be protective in these environments. So as we have cleaned up our environment, we've had this increase in immune-mediated inflammatory diseases, even beyond those that we're talking about these days. So there are a number of factors within the environment that have been associated with inflammatory bowel disease, and also somewhat with differences between ulcerative colitis and Crohn's disease. We've heard repeatedly over the past couple days about cigarette smoking. Smoking protects against ulcerative colitis, but it makes Crohn's disease worse. And there are similar features in rheumatoid arthritis, where smokers have a worse course, and actually in autoimmune thyroid disease. Smokers get Graves' disease, non-smokers get Hashimoto's thyroiditis. So another example of similar organs where an environmental factor such as smoking can actually change the direction of the immune events. All of these diseases are more common in the colder climates of North America as well as the colder climates in the southern hemisphere, less so in the tropics, whether this is due to refrigeration, whether this is due to more processed food than natural food, or whether this is related to exposure in young individuals to infections, remains to be established. That gives consideration of vitamin D, and we see the same thing across these diseases. In patients with active inflammatory disease, vitamin D is low, and it's better in patients in remission or those without the disease. But to my knowledge, that there has not been a single study that has demonstrated that adding vitamin D to vitamin D deficient patients actually improves the disease on its own. And what we're actually finding in our series, Adam, and I'd be interested in what you're seeing in your program, because you're replacing vitamin D. It's, again, one of our quality indicators. We're measuring it in everyone, and we're replacing it. What we're finding is that active disease actually impairs the ability to increase the vitamin D levels. So I think that it's probably an egg and not the chicken, but it remains to be established. So as we see inflammatory bowel disease evolve around the world, this really gives us the opportunity to look more at where the disease is currently evolving. And it's exactly the same in Asia as it was in the cities and in North America and Europe. It's not the people in the villages who are expanding it. It's the people who are in the cities who are developing these diseases. And as these countries become more westernized and all that encompasses that, we're seeing this expansion of the disease. So in the question that I gave you, we looked at genes and genetic susceptibility. As of 2018, there are now, this is several years old, there are more than 200 genes, mutations, that have been associated with inflammatory bowel disease. But despite these gene mutations, it's actually less than 15% of the population who have, less than 15% of the patients with Crohn's or ulcerative colitis actually have one of the genetic mutations that's been recognized. So 80 to 85% actually don't have one of these mutations, which makes us think that it's obviously more than just genes in this. But what's also of interest is the genetic mutations that have been recognized in ulcerative colitis and Crohn's disease. 70% are shared with other complex or immune-mediated diseases, such as ankylosing spondylitis, psoriasis, type 1 diabetes. So we're seeing a conglomeration of these risky alleles in individuals with the immune-mediated diseases. But what's most interesting in IBD, because we're now seeing a massive increase in early onset Crohn's disease in particular. We used to not see this in kids below the age of 10, and now we're beginning to see this. And in those individuals, most of them do have one of the genetic mutations. So the younger, and in particularly the very young, are more likely to have one of the mutations than individuals who get IBD at the more typical age in the second or third decades of life. And as you can see, with some of the major genetic variations that have been recently identified, we're starting to be able to consider targeting specific therapies, because right up at the top is interleukin-23 and interleukin-12 signaling and JAK signaling are some of the potential targets for drug evaluation. And you can imagine industry is taking a look at all of these mutations and trying to identify which specific mutations may or may not be helpful. So as we continue to identify the less frequent genes, we're going to identify those that do have consequence. And I think that IL-23 is probably one of the first examples leading from gene discovery onto effective therapies. And then, of course, the immune response, which is varied in individuals with these immune-mediated diseases and inflammatory bowel disease. So these are complex diseases. And again, I would predict that a word map such as this that would be applied to RA or psoriasis would look very similar with the variety of different potential targets that have been identified by genetic analyses in these individuals. So IBD, similar to our other siblings or our cousins of rheumatoid arthritis and psoriasis and the other IMIDs, are complex diseases. We actually heard Friday from Joel Pico about epigenetic changes. So it's not just our genes, it's the potential of our genes actually being modified by microRNAs, and those microRNAs can actually be passed on from parents to siblings to impact on this. So it's not just pure DNA that's doing this. There's a contribution of how the DNA is modified by epigenetics. And then how do our immune systems interact with our intestinal microflora? We know that in patients with active disease, and I think it's probably going to be the same for IBD, which is intestinal, but probably for other IMIDs, is that the diversity of the microbiome is reduced in patients with active inflammatory bowel disease. And then when their disease remits, the diversity comes back towards normal. We, again, do not know whether this is a chicken or the egg. And I've talked to some investigators who are talking about fecal transplants, in the setting of inflammatory bowel disease, and they show a picture of a forest fire, and then the dead forest, and then repopulating it. Well, that may be okay if you've got a dead forest, but if you've got active inflammation and an ulcerated gut, and you put in a probiotic or a transplant, the fire is still burning. And so we don't know whether that's actually going to have an impact. Perhaps it might be better as a maintenance agent than as an inductive agent, but as I said, we are in our infancy of studying the microflora in these diseases. And then a variety of different environmental factors, including smoking in our disease, in IBD, NSAIDs are a predisposing factor. Children who are exposed to antibiotics are more likely to get inflammatory bowel disease. Lots of interest, but really not strong data regarding the diet. And then finally, as you're going to be hearing this evening and such, the issues of stress, sleep, and general health. Individuals, if you ask a patient what triggered your flare-up, they're often going to say stress. But stress is very complicated in simplistic terms. A divorce, it may be stressful for one and unstressful for another, yet it's the same, quote, signal that we're looking at. So these are complex diseases, to say the least, that lead eventually into this chronic intestinal, chronic inflammation in these diseases. So through the years, we've recognized that T cells are important drivers of the adaptive immune response. The antigen-presenting cells, which are prevalent within the intestine, they're called dendritic cells, and dendritic cells stick up through the epithelium, which is one layer thick within the gut that separates the gut from the underlying immune system. These dendritic cells sample the molecular patterns that are present within the microbiome within the gut. And depending on the nature of the molecular pattern, sends downstream signals to either Th1 or what used to be Th2 and now are Th17 cells, or T regulatory cells to help either control inflammation or help rebalance it in the setting of regulatory T cells. Now, what's interesting is that one of the common pathways of IBD, and is what we've heard in psoriasis and psoriatic arthritis, is the interleukin-17 pathway. Indeed, ulcerative colitis is considered an IL-17 type inflammatory disorder. Yet, when you inhibit IL-17 in psoriasis, you get phenomenal results. When you inhibit IL-17 pathways in ulcerative colitis, you actually get worsening of the disease. and we've learned somewhat how there are differential effects of interleukin-17, and probably related to epithelial repair within the gut. That's an important property of this, because there's IL-17 in epithelial cells as well. So it may account for some of the paradoxical effects that we're seeing. And I think we'll really continue to learn about what are the discrepancies and what are the similarities between our diseases. So obviously, TNF-alpha is very important, and interleukin-23 is very important. But there are a number of drugs that are used in rheumatology or in dermatology, such as abatacept, which is an effective drug in RA, yet when it was tested in ulcerative colitis and Crohn's disease, actually had harmful effects. It was worse than placebo. So as we begin to dissect some of these discrepancies and similarities, why are all of these diseases increased in prevalence in patients who are receiving the checkpoint inhibitors? So we have a lot to learn yet about the similarities and differences. There is, so the cytokines are part of the adaptive immune response to individuals. We're also learning a number of the innate response potential triggers and starting to target such things as toll-like receptors, et cetera, to potentially impact on inflammatory disease in IBD. So this is a recent, Silvio Denise, who's been chairman of the European Crohn's and Colitis European Crohn's and Colitis Organization, has put together a series of these galaxy slides over the years showing how some of the drugs starting in phase 1 are moving into finally drug approval. And this is continuing to happen, but look at the spectrum of agents that are currently being explored through the different phases. There's a simple problem to this. We don't have enough patients to study all these different agents. So we're starting to have to prioritize based on very limited data, and I'm going to show you something about that. So there has been many overlaps in the similarities between the drugs that we're using across the immune-mediated diseases. Within the TNF inhibitors, I just want to point out the one question about etanercept. Etanercept was tested in Crohn's disease at the same dose that it was tested in rheumatoid arthritis, and it was a negative study. But I have to tell you, had we tested infliximab, adalimumab, certolizumab, golimumab in the same doses that are effective in other diseases, they would have been ineffective as well. And most recently, when we're talking about IL-12/23, the 45 to 90 milligram dose of ustekinumab in psoriasis is starting with 6 milligrams per kilogram in Crohn's disease. So that's 6 times 80, 500 milligram doses compared to 90 milligram doses in the psoriasis. So I think etanercept probably would have had potential in IBD, but it's just underdosed. And some of the positioning of our anti-TNF agents for IBD are frankly related to some of their dose responses at currently marketed doses. Because IBD is the small market for all of these agents. RA, psoriasis are much bigger markets. So when you turn out to have twice the dose in IBD, it's going to be a challenge. Tofacitinib is another example. The current dosing in rheumatoid arthritis, 5 milligrams twice daily, has a pricing that's potentially comparable to biologic agents. We're going to be using 10 milligrams twice daily in ulcerative colitis. So we're anxiously interested in awaiting how the pricing of these different drugs is going to change over time. So we've had a lot of success over the past 5 years or so with drug development in inflammatory bowel disease, but I want to point out just one non-success, and that's a drug that was published as a phase 2 study in the New England Journal, Mongersen, which it's an antisense compound that was administered orally that inhibited SMAD7. So again, within the inflammatory pathways, a lot of rationale for inhibiting this agent, which would prevent downstream signaling. The phase 2 study published in the New England Journal was based on clinical data alone, without biologic data of endoscopic healing. And when Mongerson was then purchased and went into phase 3 studies and eventually incorporated these more deeper biologic parameters, the drug failed in phase 3 studies. So the importance of treating beyond symptoms, as we've talked about over the past several days, and treating to biologic markers is going to be exceedingly important. So what's actually hitting the clinic? Well, we've been focusing on cytokines over the past several years. The IL-12 and IL-23 pathway that you've been hearing about over these past several days has become very relevant in the setting initially of Crohn's disease and potentially and probably in the setting of ulcerative colitis as well. We've learned that this pathway becomes upregulated early in the setting of inflammatory disease, which brings out a very important point. If you look at the cytokines that are present early in untreated disease versus late or burned out disease, they're considerably different. And so there may actually be an evolution of the different cytokines that need to be modified in different phases of the disease, and may also account why we see a duration effect on efficacy. The longer the duration of the disease in Crohn's disease, the less likely our current therapies are going to be effective in this. And then as the disease progresses over months and years, more expression of interleukin 23. And interleukin 12 is also present within chronic intestinal inflammation. So there are a number of targets, but it's going to be important where we are actually directing these targets and at what point in the disease activity. But focusing on IL-12/23, and again, we've talked about this in different realms, there was rationale for targeting interleukin 12 in the setting of inflammatory bowel disease and Crohn's disease because of those early studies. It wasn't till later that interleukin 23 was actually identified. And as you now know, IL-12 and IL-23 share one of the receptors, the P40 receptor, that is present on both IL-12 or present for both IL-12 and for IL-23. So ustekinumab, which inhibits the P40 subunit, inhibits both IL-12 and 23. We've seen its data across in psoriasis, now in Crohn's disease. Looks to be a very effective and a very safe drug. As with all of our agents, if it's given as the first therapy, it's going to be better than if it's given as the second or third therapy. But there are a number of other drugs that are under development now that are targeting the P19 subunit, which is going to inhibit IL-23 alone without any impact on IL-12. And in a comparative study, one of the IL-23 inhibitors looked initially to be potentially better than the combination of IL-12 and IL-23 in tandem. So we're going to need to see how this begins to sort out. In the Crohn's disease studies called Unity 1 and Unity 2, the most important thing to point out besides the efficacy is that Unity 1 patients had been previously exposed to TNF agents. Unity 2 patients had not. And Unity 1 were also longer duration disease. So you can see the absolute response rates are almost double. And we've seen this with every switch between agents. Once you switch from one agent to the next, you lose about 50% of the overall efficacy, which is why Adam is so adamant about doing prospective monitoring so that we get the best of each one of our agents before we switch. Because we know that when we switch, whether it's from within class or out of class, we're going to take a hit. In the studies looking at adalimumab in patients who had lost response to infliximab, it was positive, but the results were about half, approximately half, of the patients treated with adalimumab as a first TNF agent. Our first love is our deepest, the first cut is the deepest, and I firmly believe this. Okay. This is just data from two other IL-23 inhibitors that utilize the target of the P19 subunit. This is an agent that's in clinical trials, and we see similar results. And finally, a third, Risankizumab, that is also approaching marketability, demonstrated benefits in the setting of Crohn's disease, as we've seen in psoriasis as well. Vibeke is going to talk extensively about JAK inhibitors. So I think I'm just going to breeze through this rather quickly to get to the end. You're going to talk about the different JAKs? I'm sure you are. Okay. So when I know JAK, and I know Vibeke knows JAK better than I know JAK, so I'll let her talk to you in detail. Nevertheless, we have studies in inflammatory bowel disease now with tofacitinib, tested at 5 and 10 milligrams in ulcerative colitis. This is the same kind of population we see redundantly in clinical trials for drug approval. Moderate to severe disease, not responding to steroids, immunosuppressives, mesalamines, or TNF inhibitors. And again, when you look at the results in the bio-naive versus the bio-exposed, the bio-naive do better, and the higher dose actually does better in the bio-exposed patients than the patients who are bio-naive. So we're getting to see, some rationale of how we might be dosing tofacitinib according to their background history, certainly in the different settings of induction and maintenance. And the FDA just had a hearing on tofacitinib as to whether or not the 10 milligram dose should be able to be approved for maintenance as well as the 5 milligram. As we heard in that last lecture, as far as side effects are concerned, there are some dose-dependent side effects with tofacitinib that go for the 10 milligrams is twice daily is associated with more zoster infections than the 5 milligrams BID. But again, I point out, if you look at prior TNF exposure versus the non, much better results in the patients who had tofacitinib as the first agent after steroids or immunosuppressives. So we have a number of different targets. We have too many targets, frankly, by the way. The number of targets also will tell us why we're only getting 30 to 40% efficacy in any of these drugs. One, we're using them too late. We should be using them, in my view, before steroids rather than after steroids, because they are safer than steroids. But inflammation is plastic. And when we inhibit TNF, there is circuitous routes around it, so we clearly see patients who are primary non-responders to TNF and patients who lose response despite having good levels of the drug who won't respond to another in class. So the plasticity allows inflammation to pass through other mechanisms. So how are we going to actually position these drugs? Well, we're going to need to consider a lot of different factors. The disease factor, the patient factor, where this falls into other strategies, what have they responded to, what have they not responded to, and how risk averse the individuals are. If you've got in IBD, where we have young patients, males and females, anticipating pregnancy, and we're going to hear about that later. Do we want to give a drug that may potentially impact on that? What about the patient who comes in whose sister had a lymphoma? That's going to drive their decision-making. At the moment, we do not have comparative effectiveness studies. There are some underway in IBD now. We are seeing studies of ustekinumab now versus adalimumab and vedolizumab versus adalimumab. There's a reason that the comparators are looking at adalimumab. One, it's got the biggest market share at the present time, but the data don't look as potentially, it looks more challengeable than some of the others. But potentially we can go on to network meta-analyses, versus placebo, but the trials are changing. The infliximab studies were done in the late 1990s and the early 2000s, and now we're adding in endoscopic appearance. So you really can't really discuss or compare between these different trials. So many different patient factors associated with this. As far as evidence is concerned, I think we've, through these days, identified the impact of our 3 biologics now, anti-TNFs, the anti-integrins, and ustekinumab. Ustekinumab is being tested in the setting of ulcerative colitis, as are the other IL-23 inhibitors. So my last slide is, what have we learned to this point? And I've shown this before. We know that IBD, both Crohn's and ulcerative colitis, are chronic and progressive. We know that the symptoms don't reflect the inflammatory burden, and hence the FDA is requiring now evidence of endoscopic healing in addition to clinical symptoms for drug approval. We've shown, as Marla Dubinsky said yesterday, that when we treat to therapeutic targets, we get better outcomes. We've learned that the earlier you treat the patient with an effective agent, the better the absolute response rates. As Adam says, you get the most out of our initial therapy, and we are learning increasingly that the PK-PD differences across these separate some of the impacts from IBD from our other immune-mediated diseases. So, I went a minute long. I'm sorry, Adam.
That's okay. You're allowed.
Thank you.